Hello, my name is Anika, and together with Kevin and Anel, we will be presenting on Microbiome.jl and BiobakeryUtils.jl. Before we dive into the packages themselves, we first need to understand a little bit about the microbiome itself. A microbiome is a community of microorganisms living together in a habitat. In our lab, we are specifically interested in the human microbiome and how it plays an important role in human health and disease. There are many different approaches to studying the microbiome, one of which is metagenomic sequencing. Metagenomic sequencing is the analysis of all genes in all organisms present in a given microbial sample, and this generates a huge amount of data. Two of the different types of microbial community data that metagenomic sequencing generates are taxonomic profiles, which describes what microbes are present, and functional profiles, which describes what these microbes can do. Given the large amount of microbial community data generated by metagenomic sequencing, developing software tools to analyze this data is essential. For example, the BioBakery is a suite of command line tools used to profile and analyze microbial communities. Neatdata, Metaflan, and Human are three tools within the BioBakery that serve as quality control and create taxonomic and functional profiles. Moving on to our own packages, Microbiome.jl, which Kevin will go through in a moment, facilitates the analysis of microbial community data by making data more manageable, organizable, and readable. It does so by providing concrete types to store information about collective microbial communities. And just to note, Microbiome.jl is compatible with many different microbial community data types, not just for metagenomic sequencing. As you are going to see in the tutorials, the microbiome sample, taxon, and gene function types describe the metadata, taxonomic data, and functional data of individual samples and microbes. These three different types can then be combined into a larger community profile. Of note, the community profile data structure is compatible with tables.jl, allowing Microbiome.jl to link in with the JuliaStats ecosystem. We've also recently added a new metabolome type, and you can add your own additional features as well. And lastly, BiobakeryUtils.jl, which Anel will present more on, works on top of Microbiome.jl to provide a Julia interface for the command line BioBakery tools. It does so by integrating BioBakery outputs from Metaflan, Human, and Neatdata into Microbiome.jl functionality.
Hi, my name is Kevin Bonham, and as Anika said, I'm going to take you through the Microbiome.jl tutorial that we prepared for JuliaCon. So in this tutorial, we have some information for how to download some example data, how to load it into a data frame, and then convert it into a type that Microbiome.jl can understand. But I'm going to sort of skip through some of that boilerplate and jump straight to the community profile type. The community profile type consists of three main components. The first is a matrix that contains the relative abundance of individual features in individual samples. A list of those features, these can be microbial taxa or gene functions, and a number of other types. These represent the rows of the matrix. And then finally, there is a list of biosamples representing the columns of the matrix that basically refer to where these microbiomes came from. We also have the microbiome sample type. So these minimally consist of a name, but they also contain a dictionary from dictionaries.jl that can store an arbitrary amount of metadata. So here I have an example of a microbial stool sample. It has this alphanumeric name, and at this stage it doesn't have any metadata associated with it. But from this file type, we can see that this came from a stool sample, and so we can insert this using the dictionaries.jl function set or insert, and insert into that stool sample a metadatum referred to by a symbol, and then in this case a string, but the metadata values can be of any type. But I also want to talk about working with these community profiles. So we can pull out the list of features, we can pull out the list of samples, again with their attached metadata, and then we can also index into and slice these community profiles using numbers, but also using strings or regular expressions that will match on the names of features or samples. So here I'm using a row index with this regular expression. You can see it pulled out 3 rows. Microbiome.jl also contains a number of functions for calculating some useful ecological metrics. So here we have built-in Shannon index and Gini-Simpson index. And in addition to having the non-mutating form that returns the values for each column, we also have these mutating forms that will automatically add those metrics to the sample metadata, which can be really convenient. One other thing I wanted to mention about the sample metadata is that we can add them in bulk. So here I'm showing an example where I've built a data frame. It has one column that provides the name of individual samples, and then two columns of different types of metadata. And we can use this convenient set function, passing the community profile as the first argument and the metadata table as the second argument, to automatically attach all of these metadata to the proper sample, and then we can pull them back out again as a tables.jl compatible table using the metadata function. And if any of the samples are missing some piece of metadata, they don't have it recorded, those will come through in the output table as just missing values. And then we can pull out a vector of these metadata using get, which is again from dictionaries.jl. So given a community profile type, again, we can integrate with other Julia packages. And in this tutorial, I have an example using the distances package, the multivariate stats package, and the plots package to do a principal coordinates analysis and coloring the plots by different types of metadata, including the body site and the Shannon index of diversity. So I hope that's informative. Again, there's lots more information on the tutorial and in the Microbiome.jl documentation.
Hello, my name is Anal, and I'm going to be talking about BiobakeryUtils.jl and walk you through how to interact with microbial community data using this package. So, two main components of BiobakeryUtils are that it has different functions that corresponds to BioBakery command line tools that work from within a Julia session. And the second component is that it has interfaces that load these file types that are from BioBakery and turns them into Microbiome.jl types, like community profile, which serves different purposes for someone who wants to analyze these data. So to start with the command line tools, if you're already familiar with BioBakery and have it installed, all you need to do is make sure that it is in your path environmental variable. But if it is your first time, you can run BiobakeryUtils.install-deps, which currently uses conda.jl in order to install BioBakery. So each command line interface function is designed to mimic the corresponding command line tool in BioBakery and run it within a Julia session. So for example, if we want to run this need data file that could usually be run from a command line, we can use the BiobakeryUtils.jl function instead, which is metaflan, which also takes the keywords arguments that are run within the command line and take it as the keyword arguments, basically mimicking it, but from a Julia session. And we're also able to get the same result. Now on to the second component of BiobakeryUtils.jl. There's different functions that are designed to successfully load output files of BioBakery tools into Microbiome.jl types. So for instance here, the first function that we're going to look at is metaflan profile, which loads this file that was from metaflan single sample, and it loads it into a community profile that is classified within its taxons and abundances. If we want to renormalize our table into relative abundance, we could use human renorm table from the command line, or we could call human renorm from the Julia session. Lastly, we can regroup genes to other functional categories. Human typically generates profiles using uniref, but if you want different categories like ACs or KOs, we can use human regroup in order to get whichever gene type we would like. So thank you so much for listening. If you are watching this live and have any questions, you can stick around and we will be there to answer them. Yeah, so thank you.